Hi everyone. This lecture is going to cover Chapter 17. We'll discuss the digestive system. 17.1 begins with general characteristics of the digestive system. Let's define digestion. Digestion is the mechanical and chemical breakdown of foods into forms that cell membranes can absorb. This includes mechanical digestion and chemical digestion. Mechanical digestion breaks down large pieces of food into smaller ones but does not change the chemical composition, whereas chemical digestion uses enzymes to break down food particles by changing them into simpler chemicals. To summarize, mechanical digestion includes chewing and the grinding motion of your stomach. Chemical digestion will be seen when the pancreas and the enzyme, among other organs, secrete enzymes that chemically break down food into particles that can be absorbed. The digestive system has organs that carry out mechanical and chemical digestion. They also do ingestion, propulsion, absorption, and defecation. The digestive system consists of the alimentary canal along with accessory organs. The alimentary canal we'll see goes from the mouth to the anus. The accessory organs are not part of the alimentary canal. The alimentary canal is one continuous tube. However, accessory organs will be located near the alimentary canal, and they will secrete substances such as enzymes that aid in the digestion and absorption of nutrients. In summary, the digestive system consists of two portions: the alimentary canal, which is the food passageway, and that will be the mouth, pharynx, esophagus, stomach, small intestine, large intestine, and anal canal. As you can see, it's a muscular tube about 8 meters long, about 24 feet. And as mentioned earlier, accessory organs consist of organs that empty secretions into the alimentary canal. Food does not pass through them because they are not a part of the alimentary canal. These include salivary glands, liver, gallbladder, and pancreas. Figure 17.1 shows the organs of the digestive system. You can see that the alimentary canal starts with the mouth, and you can trace it down the esophagus to the stomach, to the small intestine, and then the large intestine, the rectum, and the anus. It's one long tube. Fun fact, food does not actually enter your body properly until it is absorbed into the bloodstream. The accessory organs are located near the alimentary canal, like the liver and the gallbladder, and though the pancreas is not pictured here, if you were to lift the stomach, the pancreas is deep to the stomach. It is also an accessory organ. The publishers mention that salivary glands are accessory organs. They secrete saliva, which helps to moisten your food as it creates the bolus which you will swallow. And there are also enzymatic secretions such as amylase secreted by the saliva. This is the alimentary canal separated from the rest of the body into its respective parts. You can see the mouth, stomach, small intestine, large intestine, rectum, and anus. As we learn about the small and large intestine, I want you to remember this picture. It's not the length of the intestine that makes it small or large. It is the width of the tube. The small intestine has a narrower tube than the large intestine, which is where it got its name. This is a very important component of the alimentary canal, the wall, because as I mentioned, food is not in your body until it is absorbed into the bloodstream. In order to be absorbed in the bloodstream, it has to go through the wall. And so to protect your alimentary canal really boils down to protecting this alimentary canal wall so we can keep it healthy and we can get proper nutrient digestion, and absorption. So let's take a look. The wall of the alimentary canal is composed of 4 layers. From innermost to outermost, these layers are the mucosa, the innermost layer, the submucosa, the muscularis, and then the serosa, which is the outermost layer. Let's go back to that innermost layer, the mucosa. As you could guess, the mucosa is a mucous membrane. It is folded in some areas to increase surface area. The mucosa absorbs dietary nutrients and secretes mucus and enzymes. Superficial to the mucosa will be the submucosa. This is a connective tissue layer. This nourishes cells and transports absorbed food molecules. It is generally going to be made of areolar connective tissue. Superficial to the submucosa will be the muscularis externa. This is that smooth muscle tissue. It will contain circular and longitudinal layers. This moves food and materials through the tube, and it does so through peristalsis. It can also do segmentation, for instance, in the small intestine, to further mechanical digestion. And then lastly, the outermost layer is the serosa. This is going to bind the alimentary canal to the body cavity. There will also be a serosa layer that is directly attached to each organ in the alimentary canal. In between the visceral serosa, which is connected to the organ, and the parietal serosa, which will be connected to the abdominal cavity, there lies serous fluid. If you recall, serous fluid eliminates friction. As these organs are moving, they heat up. Just like if you rub your hands together, friction will occur between your palms and fingers of the opposite hands as you rub them together. And so to prevent the heating up of your organs in the alimentary canal, the serosa fluid will keep the temperature buffered. Table 17.1 lists the layers of the wall of the alimentary canal and is a wonderful resource for you to review. Figure 17.3 shows the structure of the wall, and we'll take a moment here to look at the mucosa versus the serosa to remind you that the mucosa is that inner lining. Remember that the inner lining of hollow organs is composed of epithelial tissue, and it can vary as we move down the alimentary canal, but just remember that we call this the mucosa. It is a mucous membrane. Mucous membranes are open to the outside world, and the alimentary canal is composed of your mouth all the way down to the anus. So if you think about it, this tube is open to the outside world. Your mouth is open to the outside world, and when the anal sphincters are relaxed, so is your anus. And so the mucosa must remain moist at all times, and it must do that by secreting mucus, seen in a goblet cell here. There will be healthy microbiome of bacteria to keep the wall of the mucosa free from pathogens. And there will also be enzymes secreted by the mucosal wall as the food passes through the alimentary canal. Epithelial cells are very good at secretion. One thing that we'll look at too as we go further in the chapter is extensions of these epithelial cells. The folds in the digestive system are called such folds, and then on those folds will be finger-like structures called villi, and these are going to be projections of the folds that will increase surface area. When we look even further, each of those folds will have the epithelial cells, and the epithelial cells in turn will also have foldings called microvilli. You can see that up here in the top right picture. So all this increases surface area greatly so that you can get maximum digestion and absorption of your nutrients. We'll talk more about that later. Going superficial from the innermost mucosa we're moving out to the submucosa. So this is actually moving towards the body, the surface of the body, away from the inner lining. And this is going to be composed of the areolar connective tissue, because that is what's going to help nourish the cells of the epithelium in the mucosa. And then part of the submucosa will also contain collagen fibers, because as you recall, collagen fibers help to bind and support all the tissues in the body. Moving to the next superficial layer, we have the muscular layer. And this is the muscularis, and it is composed of circular muscles and longitudinal muscles. In your stomach, you even have a third layer called a diagonal muscle. And all of these help to propel food and substances through the alimentary canal as they move from mouth to rectum and then are expelled from the anus. The serosa is going to bind the organs to the body cavity. You can see an example of that here with the mesentery. This is the serosa membrane that will bind the intestines and the stomach to the wall of the abdominal cavity. There are two layers to serosa. Don't forget, there's the parietal layer, which is that outer layer that we see here with the mesentery, and then the visceral serosa is going to actually connect to the muscular layer, for all intents and purposes, of the organ here. There are two types of movement in the alimentary canal. There'll be mixing movements and propelling movements. As mentioned earlier, mixing movements are going to help aid in mechanical digestion. An example of that would be segmentation. I think of the small intestine kind of chewing the food in a way. That's what it does, really. And then in the stomach, remember, there are 3 layers of muscles. And so this is called churning, because the muscular movements propel the fluid in the stomach in all directions, kind of swirling it around and making sure everything is going to be mashed and bathed in that stomach acid. And so, to summarize, as you can have guessed, mixing movements do not move materials in one direction. To do that, we need propelling movements, and these move materials in one direction. This is super important, and you have sphincters along the way that will help aid in this propelling movement. Peristalsis is the name for the propelling movement, and a ring of contraction progresses down the tube and propels food particles down the tract in a wave-like motion. I picture having a sock, and you grab the sock with your left hand, and with your right hand, you squeeze the sock and then run your hand down that sock. If you noticed, the part of your sock that you're still holding with your left hand is going to elongate, while the part that you are squeezing and moving your right hand down will become compressed. And so it's this movement of elongation and compression waves that will move your food and beverage and other substances in a one-way direction down the alimentary canal. If the sphincters fail, we all know one case when that happens, and that is called heartburn. So the stomach acid is going to come back up into the esophagus, and as you can imagine or have experienced, that burns. Figure 17.4 shows the movements of the tube, the stomach churning. Look at those 3 layers of muscles go as they are sloshing and churning your food materials into what's going to be called chyme. We'll learn that word shortly. And then as the materials are very strategically emptied into the intestine, you can see the alternating rings of contraction as materials are being moved along the digestive system, the small intestine in this case. In order for all of this movement and secretion and sensation to occur, there has to be innervation. Remember, innervation means that there are nerves present. Branches of the sympathetic and parasympathetic divisions of the autonomic nervous system will extensively innervate the alimentary canal. You're going to continue to learn about the nervous system as you progress through the rest of these chapters. In fact, you're going to probably end up learning more about the nervous system as you move through these organ systems because you will review them over and over again. This is a central theme of the way that muscles move and glands secrete. And also, you know that autonomic means that we don't have to think about it. Does our emotions play a role? Of course. Stress can affect digestion, as we know, because sympathetic control takes the focus off of digestion and into your fight or flight. So in order to have healthy digestion, we want to stay in this parasympathetic division. This is what will stimulate proper and healthy digestion. So let's take a look at the plexuses that are going to control the processes in the alimentary canal. We have the submucosal plexus, which will control secretions. Remember, that's going to be superficial to the epithelial cells. So it'll be superficial to that mucosal layer. And so that's going to control the secretions that are occurring within that mucosal layer, like the digestive enzymes and mucus, etc. Then there is the myenteric plexus. This controls gastrointestinal motility. Myo is muscle, so myenteric plexus is going to be the muscle, and enteric is the alimentary canal, so the muscle of the alimentary canal. Then we have autonomic control of digestive activity broken down into parasympathetic impulses versus sympathetic impulses. So as mentioned, parasympathetic impulses will increase the activities of the digestive system, whereas sympathetic impulses will inhibit digestive actions. Lastly, on this slide, we see enteroendocrine cells. Entero, again, is going to be the alimentary canal, and endocrine will be hormones. So enteroendocrine cells are found in the stomach and the small intestine. They secrete hormones to regulate GI organs and processes. So the alimentary canal is sometimes called the second brain. I know that it is like a brain to me because food is always on my mind. Now let's take a look at some of the organs that are part of the alimentary canal directly, starting with the mouth. The mouth is the first part of the alimentary canal, and it ingests food. The process of chewing food is called mastication. It's defined as the mechanical breakdown of solid particles and also the mixing of saliva. Mastication is very important in beginning the mechanical digestive processes. The mouth can function as an organ of speech and sensory reception, such as taste. The mouth is surrounded by the lips, cheeks, and it has the tongue on the floor and the palate on the roof. The mouth includes the oral cavity and the vestibule. When you hear the word vestibule, that's an entryway. In the South, where I live, we call it a foyer. This picture 17.5 shows the mouth. You can see the lips, and let's not forget the teeth. The teeth are imperative in mastication. We'll talk about the teeth a little bit more in depth, but while this person has got their mouth wide open, I want you to see that they are not all the same type of teeth. Mammals, of which we are, have different kinds of teeth. We've got incisors, we have canines, with premolars and molars. So this is a typical example of the mammalian tooth structure. Molars are for grinding, and incisors and canines are for ripping and shredding. Other animals have the same kind of teeth. Think about a shark. They're all triangular in shape. Think about reptiles. Think about, fish. Another interesting thing is that teeth are made of dentine, which is a different type of structure than the calcium phosphate found in your bones. Dentine and enamel in the animal kingdom is largely derived from the skin, the dermal tissue. You can even call it dermal bone in some cases. And this is why sharks can continuously lose teeth and grow more teeth, is because they're just a conveyor belt of skin, and they lose those teeth because of their predatory behavior. We lose our teeth one time and can replace them, right? We lose our baby teeth, and then we have our deciduous teeth grow in, our adult teeth. And then if we lose those, then we have to have filler, implants. And it's important because without the teeth, the jaw will start to degrade in its structure. And so implants are very important to help prevent that from happening if you do lose teeth. Hopefully insurance will pay for it, but it's a long and arduous process regardless. It's best just to not lose your teeth, but sometimes we can't help it. All right, so just real quick, cheeks are the lateral walls of the mouth. Lips are highly mobile structures that surround the mouth opening, and the tongue is a thick muscular organ that occupies the floor of the mouth and nearly fills the oral cavity when the mouth is closed. There is a frenulum, which is like a little septum called the lingual frenulum. Lingual is tongue, and this connects the tongue to the floor of the mouth. You all know this piece of flesh. You've seen it when you go and you lift your tongue up. It's the little middle septum. Papilla are going to be projections that move the food and contain taste buds, and they get larger as you go further back in the tongue, deeper towards your throat. The lingual tonsils are tonsils that are on the root of the tongue. And as you know, tonsils are a type of lymphatic tissue that will help to catch pathogens before they have a chance to enter the body. So this shows the tongue, and you can see how the papilla are nipple-like. Papilla mean nipple. And then, of course, the palatine tonsils. And notice that the papillae get larger as we get towards the throat. And then here are the lingual tonsils, which actually just look like really large papilla. The palate forms the roof of the oral cavity. There's a hard palate and a soft palate. You learned about the hard palate in the skeletal chapter because it has the palatine bones and part of the maxilla. Wherever there are teeth attached, that is the maxilla because that's the upper jaw. And then posterior to the maxilla is going to be the palatine bones. And the palatine bones are going to form the margin of the nostrils. And then behind the palatine bone, moving, inward, deeper into the mouth, you have the soft palate. And this consists of the muscular arch and ends in the uvula. And the uvula is going to function in the gag reflex. And I recall us talking about that with the glossopharyngeal nerve in the cranial nerve discussions. Palatine tonsils are going to be lymphatic masses on the side of the tongue, and the pharyngeal tonsils, or adenoids, are masses of lymphatic tissue in the posterior wall of the pharynx. I believe those are the ones that give us the most trouble. So here we see the lingual tonsils, the palatine tonsils, which are your sides of the tongue, and then let's look for those pharyngeal tonsils, which are going to be the adenoids. Okay, they're a little bit taller, so they can get all gunked up with snot. And then these ones, when you open your mouth and get those tonsillitis and stuff like that, blech, a lot of times those get in the palatine tonsils. So we got to get those out because they can get big and smelly. All right, so teeth. I know I said a little bit about it before, so we'll just kind of go through. They are the hardest structures in the body, and they're not part of the skeleton system. Remember, they are made of dentine and enamel, and so they actually kind of come from the skin. And there are all kinds of foods we can eat because we have an omnivorous tooth pattern. That's what that dentition means. We have 20 primary deciduous teeth that fall out, and then they are replaced by 32 secondary or permanent teeth. And the types of teeth are central incisors, lateral incisors, canines, first molars, second molars, and third molars. And then, of course, I think that, let's see, let's go back here. So there's the middle incisors, lateral incisors, canines, premolars, and then first molar, second molar, and then third molar. And then, of course, we have the wisdom teeth that can come in and cause problems. All right. So the teeth break down food particles to smaller pieces, beginning the process of mechanical digestion. And your tongue helps, and chewing will mix food with enzymes. The incisors are used for biting. The canines are for grasping and tearing and shredding, and the molars and premolars are for grinding. There's the crown and a root, which meets at a neck. And the enamel covers the crown, and dentin makes up most of the tooth and surrounds the pulp cavity. The periodontal ligament holds the root in place, along with cementum. So when a body dies and the organic tissue breaks down, that includes the ligaments. And so a skull is going to be fragile in terms of its teeth and its lower jaw. So the reason the lower jaw isn't connected to a skull when you find one, like a deer skull, let's keep it less morbid, right? A deer skull. Usually you'll find like just the top part of the skull, and the jaw might have fallen somewhere else or been carried away by another animal. And so there's a ligament that holds the jaw onto the skull. And so once that ligament has degraded, then that jaw, that mandible falls off. Same is true in the teeth. The teeth are going to be very loose in a skull if that periodontal ligament breaks down. So here we see the primary teeth and the secondary teeth up here. Isn't that wild? Oh my gosh. And so they'll, of course, replace those primary teeth. And we see the crown and the root and then the neck in which they meet. And then, of course, we know that they are covered with gums. And periodontal disease is the disease of the gums. It starts with gingivitis, and it's when bacteria get in there and they secrete acids and all kinds of things that decay the enamel of the tooth and end up, causing tooth loss as decay continues. Not only can it cause decay, but as the bacteria from that biofilm and that plaque break off, it can go through the body and it can cause issues such as endocarditis. This is a table 17.2 and figure 17.9 that will go through each type of teeth that we have, including the primary teeth and then the 32 secondary teeth that we have. Here's a pretty picture of the tooth. We see the enamel and then the dentin underneath, and then there's the pulp, which is where the trigeminal nerve comes in and is so very sensitive. Here you see the periodontal ligament, and remember there's a cementum in there, and then the gum is called gingiva, so that's why it's gingivitis if you have the inflammation of that, and that can lead to periodontal disease. So let's talk about that. With dental caries, there's going to be a cavity that forms within the enamel of a tooth, and a cavity is an opening, and a hollow space. It's formed when sticky foods lodge between the teeth or in crevices of molars. The bacteria on the teeth metabolize sugars, and then they produce acidic byproducts that destroy enamel and dentin. According to the American Dental Association, or ADA, the following may help prevent dental caries: brushing and flossing, dental exams and cleanings, fluoride treatments, and sealants. My mom was a dental hygienist before her retirement, and she got us sealants when we were young, and I'm very grateful to her for that. Table 17.3 is going to review the mouth parts and their functions in digestion. Now let's talk about salivary glands. Salivary glands will secrete saliva. Saliva moistens food particles and binds them together, and it dissolves food so it can be tasted and swallowed. It contains enzymes such as amylase that begins the chemical digestion of carbohydrates, and it also contains bicarbonate ions, which you know keeps pH favorable for enzyme activity and protects teeth from acidic foods. So it's a buffer. There are 3 pairs of major salivary glands: the parotid glands, submandibular glands, and sublingual glands. The parotid glands are the largest, and they secrete serous saliva with amylase. The submandibular glands are on the floor of the mouth, and they secrete serous saliva and mucus. And then the sublingual glands are located under the tongue, and they secrete mainly mucus. Many minor glands scattered throughout the mucosa of the tongue, palate, and the cheeks keep the lining of the mouth moist. The different pairs of salivary glands have varying proportions of 2 types of secretory cells. We have serous cells and mucus cells, which is good because we mentioned those in the last slide. So now we can elaborate on what serous saliva is versus mucus. So serous cells produce a watery fluid containing a digestive enzyme called salivary amylase. Amylase is going to split starch and glycogen into disaccharides. Then the mucus cells will secrete mucus, and mucus binds food particles and lubricates food while swallowing. So we think of mucus as this gross byproduct of being sick, and it only becomes a gross byproduct of being sick when you're sick, and it's trapping all kinds of bacteria and white blood cells, and it gets real phlegmy. Mucus should be just really nice and, clear and moist and a little thick, but it shouldn't be yellow or green or anything like that. So think of like a slug. I know that's gross, but a slug has mucus to keep their skin moist, so they don't dry out, and of course they leave that slime trail, but it's a clear fluid, right? Table 17.4 summarizes the major salivary glands for your review. Figure 17.11 shows the major salivary glands, and you see the submandibular gland. So sub is mandibular, deep to the mandible, that lower jaw. Sublingual gland, you can see that it is deep to the tongue. Lingual is tongue. And then the parotid gland. So this is going to be near those parotid lymph nodes that we learned about, the parotid tonsils in the previous slides. Here shows the serous cells versus the mucus cells. Notice that the mucus cells are a lot larger, and you can actually see that they look clear on the inside. As mentioned earlier, that mucus secretion should be clear, and just keep your mucous membranes moist. Moving inferiorly down the alimentary canal, we have reached the pharynx and the esophagus. We used to always say pharynx, but it's not. It's pharynx and larynx. So the R is before the Y, and the Y is before the N. Rinx. So pharynx and esophagus do not help in food digestion. Both the pharynx and esophagus have muscular walls which function in swallowing. And so when it says they do not help in food digestion, that means that their role is propulsion. So they're not secreting enzymes or, digestive juices, but they are imperative to the digestive process because they propel your food from your mouth to your stomach. So the pharynx is a cavity posterior to the mouth, and it extends from the nasal cavity to the esophagus. There's 3 parts of the pharynx: the nasopharynx, oropharynx, and laryngopharynx. Nasopharynx is posterior to the nasal cavity. It's an air passage, and it contains openings to the auditory tubes, which are your ear tubes. And actually, as you talk, your voice resonates in the nasopharynx, which is why your voice sounds deeper to you than it does to other people, because you're kind of getting a resonant, um, output. The oropharynx is going to be posterior to the oral cavity, and this is your air and food passage. And then the laryngopharynx is posterior to the larynx and is the passageway to the esophagus. So as you swallow, then the esophagus is going to take the food from the pharynx and propel it towards the stomach. Now, there are going to be some things that will help out in this because the digestive system is actually an outpocket of the respiratory system. And so this can pose a problem that we'll talk about in a moment. But first and foremost, here you see that larynx. So again, it's like Larry, not Larny, is Larry. Larynx, larynx. Pharynx, larynx. And you can see the musculature of the esophagus and the larynx. All right, so let's talk about this propulsion, and it's called swallowing. So swallowing can be divided into 3 stages. The first stage is voluntary, and the second and third stages are going to be reflexive. So the first stage, the voluntary stage, occurs when saliva is mixed with chewed food, forming a bolus. So it's just kind of like a ball of food. The second stage starts as food reaches the oropharynx, the back of your mouth. It stimulates sensory receptors and triggers the swallowing reflex. The soft palate and the uvula rise to protect the nasal cavity from the food. The hyoid bone and the larynx elevate, and the epiglottis closes off the top of the larynx to protect the trachea. Because the respiratory system, I might have said it backwards in the previous slide, my apologies. The respiratory system is an outpocket of the digestive system. So the respiratory system comes off of the digestive system. And so there is a moment where the trachea and the esophagus are connected. And so choking can occur if the epiglottis fails to close off the top of the larynx. All right, so once that occurs and we're going down the right tube, we're going down the esophagus, the longitudinal muscles of the pharynx will contract, the inferior constrictor muscles relax, and the esophagus will open, and the peristaltic waves will force the food into the esophagus. So, whoo, we made it. And now we're at that third stage, and peristalsis will transport food in the esophagus to the stomach. So in the first step, the tongue forces food into the pharynx. And then you can see in part B of figure 17.14 that the soft palate, hyoid bone, and larynx are raised, and the tongue is pressed against the palate, and the epiglottis closes, and the inferior constrictor muscles relax so that the esophagus opens. So we've closed the trachea, we've opened the esophagus, and notice that the esophagus is posterior to the trachea, and it is sandwiched between the trachea and the vertebral column. And so looking at that trachea, you can see that the respiratory system is an outpocket that evolved off of the digestive system. Once we have entered the esophagus, we've done a good job, we've swallowed correctly, and our superior constrictor muscles contract and initiate a peristaltic wave that forces food into the esophagus. And then it's all gravy from there. The peristaltic wave moves food through the esophagus to the stomach. So remember, sphincters are going to be very important in helping to regulate the movement of this food. And let's go ahead and go down to the last bullet where it says the lower esophageal or cardiac sphincter regulates food passage into the stomach. It closes to prevent regurgitation of food. So if the cardiac muscle fails, then we get heartburn. So it's not called heartburn because our heart is what's hurting. It's our esophagus, and everybody knows that. But the reason that heart is in there is because it's named for that cardiac sphincter. And I imagine the cardiac sphincter is named because it is located right there in the region posterior to your heart. So voila, we have our esophagus and then, our cardiac sphincter. And so you can imagine that the heart is right here if we're looking at the anatomic position. So right there, cardiac sphincter and cardiac muscle, the heart, right there by each other. And then we see the stomach and then, of course, the intestine. So now let's talk about the stomach in 17.5. The stomach is a J-shaped pouch-like organ, about 25 to 30 centimeters long. And I'm thinking of all the different animal stomachs that I have seen, and they all pretty much have that J shape. We did a perch dissection in class last week in my Gen Bio class, and sure enough, even the fish had a J-shaped stomach. And so there's parts to that stomach that are very important and that make that J shape. We'll talk about those shortly. But before we do that, let's look at the rugae. The rugae are folds of muscles and submucosa, folds of mucosa and submucosa that allow for distension. Distension means stretch. So the stomach can stretch to fill all that volume of food and beverage that you are cramming into it. And so the stomach will receive food from the esophagus, mix that food with gastric juice, initiate protein digestion. It has limited absorption, like it can absorb alcohol and medications and aspirin and Tylenol and all. And then it moves food into the small intestine. The stomach contains the following layers of smooth muscle: an inner circular layer, an outer longitudinal layer, and then, as mentioned before, some portions have the third innermost layer of oblique fibers. Figure 17.17 goes over the parts of the stomach. And so the literature portion talks about the portions of the stomach being the cardia, the fundus, the body, and the pylorus. So the cardia is going to be the region near the opening to the esophagus, and it's going to contain the lower esophageal sphincter. The fundus is the rounded area that rises above the cardia, and it functions in temporary food storage, which sometimes contains swallowed air. The body is the main portion, and it lies between the fundus and the pylorus. The pylorus is my favorite part. It's the distal portion, and it is closest to the smallest intestine. It is a funnel shaped, and it narrows to become a canal. So it's the funnel shaped pyloric antrum narrows to become the pyloric canal. And there we find the pyloric sphincter. This is the most brilliant thing ever. The pyloric sphincter is a circular smooth muscle, and it controls gastric emptying. So gastric glands contain 3 types of secretory cells, which produce a mixture called gastric juice. The components of gastric juice are pepsinogen, pepsin, gastric lipase, hydrochloric acid, mucus, and intrinsic factor. All of these are important, and you can see that pepsinogen and pepsin are going to be involved in the digestion of proteins. Gastric lipase will digest fat. Hydrochloric acid, or HCl, produced by parietal cells, is going to be what converts the inactive form of pepsin into its active form and digest proteins. Mucus is secreted to provide lubrication and protect the stomach lining. And then intrinsic factor is produced by parietal cells, and it is required for the absorption of vitamin B12. Some people don't produce intrinsic factor, and this can cause them to become deficient in vitamin B12. So taking vitamin B12 sublingually tries to bypass the intrinsic factor need, and also taking vitamin B12 injections. All right, so 17.5 is going to summarize the major components of the gastric juice. And then here we can see the lining of the stomach. So remember, we've got the mucosal layer, we've got the submucosa, we've got the muscularis, and then we have the serosa. All right, regulation of gastric secretion. Gastric juice is produced continuously, but the rate of production varies and is under neural and hormonal control. Neural regulation will course, have sympathetic and parasympathetic parts. And as you remember, sympathetic is going to decrease gastric activity, whereas parasympathetic will increase gastric activity. Hormones include somatostatin, which will inhibit hydrochloric acid secretion, which must be why the medicine for gastric reflux are like statins. And then we have gastrin, which is going to increase gastric juice secretion. And then cholecystokinin, which is abbreviated CCK, released by small intestine cells when proteins and fat enter the small intestine. This decreases gastric motility. And this is one of the reasons why proteins and fats keep you fuller longer. All right, so figure 17.20 shows a few steps in the regulation of gastric secretions. Step 1, impulses conducted by the parasympathetic preganglionic nerve fiber, which is from the vagus nerve, is going to be found in the stomach. Remember, the parasympathetic nerve endings are going to be actually in the organ, basically, and then the post ganglionic nerves of the parasympathetic are going to be like connected to the organs themselves. So the parasympathetic nerves are going to have long preganglionic axons and then very short postganglionic axons that are basically in the organ. That's what I'm trying to say. All right, so then number 3, impulses will stimulate the release of gastrin into the bloodstream. And number 4, gastrin stimulates glands to release more gastric juice. The cephalic phase is the first phase of gastric secretion. The sight, taste, smell, or thought of food triggers parasympathetic reflexes. Gastric juice is secreted into the response. Think Pavlov. The second phase is the gastric phase. Food in the stomach chemically and mechanically stimulates the release of gastrin, which in turn stimulates secretion of gastric juice. Reflex responses also stimulate gastric juice secretion. The third phase is intestinal phase. As food enters the small intestine, it stimulates intestinal cells to release intestinal gastrin, which in turn briefly promotes the secretion of gastric juice from the stomach wall. This phase primarily inhibits gastric juice secretion by sympathetic reflex and the hormone cholecystokinin responses. Gastric absorption. As mentioned earlier, there will be some breaking down of proteins, but not really much absorption going on in here. So the stomach can absorb the following substances: some water, some salts, certain lipid-soluble drugs, and some alcohol. Otherwise, nutrients are absorbed in the small intestine. Here we're going to talk about my favorite part, the mixing and emptying actions. The mixing of food in the stomach with gastric juice produces a semi-fluid paste called chyme. Peristaltic waves push chyme towards the pylorus, and small amount of chyme is transported through the pyloric sphincter at a time. And it's like 3 squirts a minute, something like that. So when peristaltic waves reaches a distal portion of the stomach, the contents are pushed backwards, resulting in a mixing action. The lower esophageal sphincter prevents the backflow of chyme into the esophagus. The rate of emptying depends on the consistency of chyme and the type of food, so it would be slower for, fats and proteins than it would be carbs. As chyme enters the duodenum, which is the first part of the small intestine, accessory organs add secretions to act on chyme. This is that coolest part. So remember, we're looking at the most intense acid, right? This is hydrochloric acid in the gastric juice, pH of 0 to 1. So its purpose is to convert these enzymes into protein digesting enzymes so that you can do protein digestion in your stomach and start breaking down these foods. So if the stomach secretions were to go into your small intestines would be digested by your own gastric juice. So your body is so smart. It adds secretions that are going to neutralize and buffer the acids of that gastric juice. And that's why the pyloric sphincter has to be regulated. It can only squirt a little bit of juice into the intestine at a time so that that juice can be neutralized before going into the intestine, and most of the absorption then occurs. Vomiting. This is when things go the wrong way. So this is a result of reflex that empty stomach in the reverse direction. Causes include certain drugs, toxins from foods, the overstretching of the stomach, body motion changes, motion sickness associated with the inner ears. And the vomiting center is in the medulla oblongata. Stomach movements in figure 17.21. We see the lower esophageal sphincter is relaxed and food is entering the stomach. Both sphincters are contracted during step B when chyme is being churned. And then in C, you see the pyloric sphincter is relaxed for just a moment so that a little bit of that gastric secretion can enter the small intestine. The enterogastric reflex starts when the duodenum fills with chyme, as shown in number 1. And number 2, sensory stretch receptors are stimulated. Number 3, sensory impulses are conducted to the central nervous system via the vagus nerve. And then number 4, a reduction in parasympathetic activity decreases peristalsis in the stomach wall. We're slowing down the movement of our gastric secretions into our intestines so as not to burn our intestines. Heartburn is a common problem. Stomachache results from eating a lot of food too quickly, and it takes up to 20 minutes for the hypothalamus to sense a full stomach. Excess fullness leads to abdominal pain and gastric reflux as stomach contents enter the esophagus. Stomach contents in the esophagus cause inflammation called esophagitis, and it feels like the pain is derived from the heart, so it's called heartburn. And acids can provide some relief. Prevention is to eat small meals, eat slowly, stay upright after eating, avoid caffeine, nicotine, and alcohol. Here's an accessory structure. This is the pancreas, and it is going to be mentioned here because you can see that it is going to have an endocrine and exocrine functions, right? So endocrine, we know it secretes insulin and glucagon to regulate blood glucose. You might not have known that it has an exocrine function as well. Exocrine glands are going to secrete substances onto the surface of that organ, like via a duct, and the exocrine secretions of the pancreas are going to be digestive enzymes. And so there are several types of cells, like pancreatic acinar cells, which make up most of the pancreas, and they release the pancreatic juice into tiny ducts, which lead to the pancreatic duct. The pancreatic duct, along with the common bile duct from the liver and gallbladder, empties into the duodenum of the small intestine. The pancreatic duct and the common bile duct join at a dilated tube called the hepatopancreatic ampulla. The hepatopancreatic sphincter surrounds the ampulla and controls the movement of bile and pancreatic juice into the duodenum. Figure 17.23 shows the structure of the pancreas. Pancreatic juice contains enzymes that digest carbohydrates, fats, proteins, and nucleic acids. Components of the pancreatic juice include pancreatic amylase, pancreatic lipase, trypsin, chymotrypsin, carboxypeptidase, nucleases, and bicarbonate ions. Pancreatic amylase will split the starch and glycogen into disaccharides. Pancreatic lipase breaks down triglycerides. Trypsin digests proteins. Chymotrypsin digests proteins as well. Carboxypeptidase also digests proteins. Nucleases digest nucleic acids. And bicarbonate ions make pancreatic juice alkaline and buffer that stomach acid. The regulation of pancreatic secretion is through hormones and nervous system regulation. The hormones that help regulate the release of pancreatic juice include secretin and CCK, or cholecystokinin. Secretin stimulates pancreas to release pancreatic juice high in bicarbonate ions, and cholecystokinin stimulates the pancreas to release pancreatic juice high in enzymes. And from what I understand, your pancreas can actually learn what kind of foods you most regularly eat and can secrete enzymes and hormones accordingly, which is one reason why changing your diet needs to be done slowly so that you give your pancreas time to learn your new system. So another regulation is the nervous system regulation. During cephalic and gastric phases, the parasympathetic impulses stimulate the pancreas to secrete digestive enzymes. Figure 17.24 highlights the regulation of pancreatic secretion. Number 1, acidic chyme enters the duodenum. Number 2, the intestinal mucosa secrete or release secretin into the bloodstream. And number 3, secretin stimulates pancreatic duct cells to secrete bicarbonate ions. And number 4, the pancreatic juice rich in bicarbonate ions passes down the pancreatic ducts into the duodenum. Again, I think that's just the most brilliant thing. Nature and the body is so amazing. Other accessory structures are the liver and gallbladder. The liver is the largest internal organ. It's located in the upper right abdominal quadrant, just beneath the diaphragm. It's a reddish-brown organ and is well supplied with blood vessels. The liver has 4 lobes. The largest lobe is the right lobe. The left lobe is smaller than the right lobe. The quadrant lobe is the minor lobe near the gallbladder, and the caudate lobe is the minor lobe nearer to the inferior vena cava. Lobes consist of lobules, which are functional units of the liver. They contain liver cells called hepatocytes, which are found around a central vein. Hepatic sinusoids are channels for blood between plates of cells. The hepatic portal vein brings absorbed nutrients to sinusoids. I know you've heard of the hepatic portal vein before. That's what brings absorbed nutrients from the intestines to the liver, and then it can go into the bloodstream properly. The hepatic artery brings oxygen-rich blood, which mixes with oxygen-poor blood from the portal vein. Blood flows from the sinusoids to the central veins, then to the hepatic veins. BIOL flows from the ductules to hepatic ducts and then to the common hepatic duct. Figure 17.26 highlights the lobes of the liver. Right lobe, left lobe, and then 2 minor lobes. Figure 17.27 highlights the hepatic lobules, which, as we remember, are the functional units of the liver. Figure 17.28 shows the path of blood and bile in the hepatic lobule. And so remember that the hepatic portal vein is bringing the nutrients from your intestines into the liver for, absorption into the actual bloodstream. And then the hepatic artery is going to mix oxygenated blood from the heart and the artery with the deoxygenated blood that's in the vein. Liver functions. The liver carries on many important activities. It produces glycogen from glucose. It breaks down glycogen into glucose. So it produces glycogen from glucose and it breaks down glycogen into glucose. So the insulin is what's going to stimulate it to produce glycogen from glucose. Insulin will help you to store leftover glucose that you did not use for energy. So you need to get that excess glucose out of your bloodstream and into your liver and your muscle cells as glycogen. And so the liver will do a big part of that and produce glycogen from leftover glucose that you didn't use. And then when you are hungry, the liver can break down the glycogen into glucose. And it does that in response to glucagon. And if you recall, the insulin and the glucagon are secreted by the pancreas. So another thing the liver does is it converts non-carbohydrates to glucose. And from what I understand, if you have too much excess glucose that you cannot store as glycogen, it can stay in your bloodstream, which of course is bad. We know it leads to diabetes, but it can also be converted into adipose cells or triglycerides, which are then stored in your adipose tissue. So it can lead to excess fat gain. Another thing the liver does is it oxidizes fatty acids, meaning that it strips electrons off of fatty acids and gets them ready for, energy. It synthesizes lipoproteins, phospholipids, which are the cell membranes, and cholesterol. And cholesterol gets a bad rap, but remember, cholesterol has the HDL cholesterol, which is healthy, and also steroid hormones are derived from cholesterol. The liver converts excess carbohydrates and proteins into fats. It deaminates amino acids, meaning it takes the amino groups off of the amino acids. It forms urea. It synthesizes plasma proteins. It converts some amino acids to other amino acids. It stores glycogen, iron, and the vitamins A, D, and B12. It phagocytizes damaged RBCs and foreign substances or antigens. It removes toxins such as alcohol and certain drugs from the blood. It acts as a blood reservoir, and it has a role in digestion for secreting bile. Table 17.7 shows the major functions of the liver. I read an anatomy book once that it says the liver is so important that instead of saying I love you with all my heart, you should say I love you with all my liver. Replacing the liver. This is why I didn't mention regeneration earlier. I wanted to, but I knew this slide was coming. So the liver is required to maintain life. And the liver is capable of regeneration if 25 to 30% of it is healthy. If cancer spreads to the liver, life can continue only weeks to months. A donor can donate part of the liver as regeneration can occur. There is a shortage of donor livers for transplant. Extracorporeal liver assist device, ELAD, can temporarily perform blood cleansing functions of the liver until a donor liver becomes available. However, ELAD is bioartificial since it contains the artificial portion, which is cartridges, and a biological portion, which would be human liver cells. The patient's blood plasma is passed through the ELAD, where toxins are removed and liver secretions are added, and then plasma is returned to the patient. And it's amazing how large these machines have to be that do that. Like with dialysis for the kidney, how ginormous these machines are when it's just your wee little kidney that is filtering all that blood for you all day, every day. Our body really is amazing. All right, so let's talk about the composition of bile. Bile is going to be like salts and pigments. So it's a yellowish green liquid that hepatic cells will continuously secrete. And you know that it's going to be stored in the gallbladder. And so bile salts will be produced from cholesterol, and they emulsify fats, which mean they help to break fats up into smaller components that can be more easily absorbed into the bloodstream. Only bile component that has a digestive function is the bile salts. And that's why you add salt into your water with oil, because it breaks that oil up into smaller droplets. So your body's smart, it knows that, and so it makes its own salts to break up fats for absorption. And then we have bile pigments. These are bilirubin and biliverdin. So verde is green and ruby is red or yellow. And so bilirubin and biliverdin are derived from hemoglobin, and they are going to be passed in your feces, which is why you get that brownish color to your feces. So then the bile is also going to have cholesterol in it and electrolytes. So if you have issues with your gallbladder and with bile, as you know, you have to really reduce the amount of fats and cholesterol that you consume, because it can cause the irritation and inflammation of that gallbladder. And that's not an easy surgery. And people used to say it was so easy, but from what I understand, there are some complications that can occur that we're figuring out. And so it's not just as, pop in, pop out as was once thought. So the best thing, of course, is to try to keep your gallbladder healthy and clean to prevent the need for surgery. So if there is jaundice occurring, that's the yellowing of the skin, the eye sclera, which is the white of your eye, and your mucous membranes. And it's caused by the blockage of bile ducts and diseases such as cirrhosis or hepatitis, or rapid red blood cell destruction. Hepatitis is an inflammation of the liver. There are many types, both chronic and acute, and they vary in their severity. So remember, chronic is persistent over time, and acute hits you really quickly, really fast. The most common cause of hepatitis is one of several types of viruses. Some people have symptoms and some do not. They can still be carriers even without symptoms. Some forms are blood-borne, and some forms are transmitted by contact with food, body fluids, or objects contaminated with feces that contain the virus. Antibiotics are not effective against viral hepatitis. Hepatitis A is transmitted by contact with food or objects contaminated with feces containing the virus. Hepatitis B is transmitted by contact with body fluids containing the virus. And hepatitis C is transmitted through blood contact. Half of all known cases of hepatitis. I'm going to donate blood after I record this lecture, and in today's, little "Are you healthy?" questionnaire you have to fill out, it asks if you've been exposed to hepatitis, particularly hepatitis C. And thank the Lord, the answer for me is no. All right, the gallbladder is going to be a pear-shaped sac found on the inferior surface of the liver. So again, we want to keep it healthy. And it has a duct called a cystic duct. It's the only entry and exit duct. It merges with the common hepatic duct to form the common bile duct. So this stores and concentrates bile. And it releases the bile into the duodenum of the small intestine via the common bile duct at the hepatopancreatic ampulla. The hepatopancreatic sphincter regulates the release of bile into the duodenum. Gallstones. The gallbladder normally concentrates bile salts, bile pigments, and cholesterol. Sometimes cholesterol precipitates and forms solid crystals. Crystals can enlarge, forming gallstones. Causes are excess bile concentration, too much cholesterol secreted by the liver, or the inflammation of the gallbladder. Bile release under the control of the hormone cholecystokinin, which stimulates contraction of the bladder. A gallbladder with gallstones is shown here in figure 17.29. And so again, once that gallbladder becomes inflamed, then the patient may need to have surgery if it gets really bad. So, don't let me scare you away from getting surgery at all. Like, if you need surgery, you need surgery. But again, what I was saying is you want to try to prevent it. But Lord, don't let me let you go out there and not get surgery and have your gallbladder bust, because that's really, really bad. I've had a few colleagues who that happened to, and that's almost you going like an appendix burst. So just, keep your gallbladder healthy. And if you have family history of gallbladder disease, or you're pregnant or anything like that, and, you just want to make sure that your gallbladder is healthy, then I urge you to talk to your doctor about it. All right. So the regulation of bile release by cholecystokinin. Number 1, chyme with fat enters the duodenum. Number 2, cells of the intestinal mucosa secrete the hormone cholecystokinin into the bloodstream. Step 3, cholecystokinin stimulates the muscular layer of the gallbladder wall to contract. Step number 4, bile passes down the cystic duct and the bile duct to the duodenum. And step 5, the hepatopancreatic sphincter relaxes and bile enters the duodenum. Remember, bile is going to have the pigments that are from the broken down heme, and then bile salts. So here we go. Functions of bile salts. They aid digestive enzymes through emulsification. So this is the breaking up of large fat globules into smaller droplets like a soap or detergent would. And it aids in fat digestion by increasing the surface area accessible to the enzyme lipase. And bile salts also enhance the absorption of fatty acids and cholesterol by forming micelles. And you've heard of micellar water before. And so micellar water is going to be like these little membranes that are going to take like toxins and stuff off of your skin. And so micelles, as I understand in the body, are going to help to transport fats and all of that good stuff around the body. So bile salts will also help absorb fat-soluble vitamins, A, D, E, and K, and almost all bile salts are recycled. Gallbladder disease. Okay, we just talked about all of that. And so symptoms, because you're probably asking, oh my gosh, do I have it? So symptoms: pain in the right upper abdominal quadrant, back and right shoulder. Perhaps you have nausea and sweating. Tests include ultrasonography of a gallbladder or an x-ray called a cholecystogram, because cholecysto means gallbladder. Gallstones can be found in the gallbladder, cystic duct, hepatic ducts, or the common bile duct. And again, anybody who's had them, it's been painful. Treatments: cholecystectomy, surgical removal of the gallbladder, or an endoscopy to find stones or remove them from certain areas. All right, table 17.8 is going to review some hormones of the digestive tract. But let's go on into the small intestine. All right, this is where the absorption happens. It is going to be a tubular organ that extends from the pyloric sphincter to the beginning of the large intestine, and it fills most of the abdominal cavity. So don't forget, small intestine doesn't mean it's short and little. It means it's narrow. It should be the narrow intestine, but it is long. It is going to make up most of the abdominal cavity. And from what I understand, if you were to iron out the intestine and spread it all out with all the folds and villi and microvilli, you could cover like a singles tennis court or something like that with the surface area of your small intestine. It's huge. And it's supposed to be because it completes digestion of the nutrients in chyme, and it absorbs the products of digestion, and it transports the remaining residue to the large intestine. So we give a lot of focus on the stomach and like the colon when we talk about digestive health, but keeping the small intestine healthy is imperative for proper digestion and absorption of nutrients. And it's something that I think about every day of my life because I love food and am always wanting to eat food, and I don't always want to eat the best food. And then some foods upset my stomach. And anyway, I'm just really, really focused on trying to heal my small intestine. And the three parts of my small intestine that I'm trying to heal are the duodenum, the jejunum, and the ileum. The duodenum is the shortest and most fixed portion of the small intestine. It's right there where that pyloric sphincter enters the intestine. The next portion is the jejunum. It's the middle portion, and it's thicker and more active than the next portion, which is the ileum. The ileum is the distal portion, and it contains lymph nodules called Peyer's patches. So the jejunum and the ileum are suspended by a double layer fold of peritoneum called a mesentery. And remember, the peritoneum is the name of the serous membrane of the abdominal cavity. So I'll repeat that. The jejunum and the ileum are suspended by a double-layered fold of peritoneum called a mesentery, which transports blood and lymphatic vessels and nerves to the wall of the intestine. A double fold of peritoneum called the greater omentum drapes down from the stomach over the large intestine and the folds of the small intestine. So everything is very well organized and supported here in your abdominal cavity. We can see our mesentery again, connecting to that small intestine, and then there's your mesentery a little bit more spread out. When we do the fetal pig dissection in the actual class, it's so cool because the small intestine of the pig is like balled up, like a little ball, but all the portions of its small intestine are attached to this mesentery, and so you can't move it. Like, it's fixed in there in the abdominal cavity. And here's a side view showing the mesentery and how it just wonderfully, gorgeously holds the intestines in place. I always wondered as a kid, how come my intestines don't just, fall to the floor of my abdomen, my belly, right? How are they, standing up like they are? And it's because of that beautiful mesentery. And then, of course, bringing blood vessels and protecting the small and large intestine anteriorly, just deep to your muscles there, is the greater omentum. All right, let's visit villi and microvilli. I already gave you a peek into what that is, so now the literature will elaborate. So intestinal villi are tiny projections of the mucosa, and they greatly increase the surface area for the absorption of digestive end products. Each villus consists of simple columnar epithelium with a core of connective tissue. There will be blood vessels and lacteals in each villus. The blood vessels absorb most nutrients, and the lacteals are a part of the lymphatic system, It's a lymphatic capillary that absorbs large fats, and it also has, of course, there's nerve fibers in the villus. So the blood vessels absorb most nutrients, the lacteals absorb large fats, and then, of course, you've got nerve fibers. Free surfaces of epithelial cells will contain microvilli, which also increase the surface area for absorption. So you have the folds of the small intestinal wall, and then you have the fingers that are on the folds of the small intestinal wall, and those fingers are called intestinal villi. And then all along the intestinal villi are going to be the epithelial cells, and extensions of those epithelial cells' plasma membranes are called microvilli. Intestinal glands are located between the bases of the villi, and plicae circularis are circular folds of mucosa, which also increase surface area. Those are those folds that I just mentioned. All right, so the lumen, I love this word, lumen. It's like illuminate. There's light going through it. And so lumen is going to be that center of the organ. And so where the lumen is, that's the middle of the intestine, if you would, where the nutrients are flowing through. And of course, we have the folds, and then we have the villi. See those finger-like projections. And here is a villus in detail, and all along the villus will be your simple columnar epithelial cells, and on the tips of each of those will be microvilli. Running up the core of the villus is going to be your lacteal, which is going to absorb large fat droplets, so it'll be milky in its consistency and its color. It'll be white, which is why it's called a lacteal, because it's like milky, lactase, milk, you know, lactose. And so then we have the blood vessels, the capillaries as well, that are going to absorb nutrients other than fat. So here is your columnar epithelial cell with the microvilli attached. And so the microvilli are extensions of the plasma membrane itself. I like these vesicles here. So this is showing like how the food gets into your blood, which I hope it elaborates on in a minute. But as you can see, this is where the lumen would be, where it says permission required for reproduction or display. That's the lumen. That's the middle of your intestine there. And so imagine that this red laser pointer is going to be the nutrition absorbed from your diet. So it's actually going to be absorbed in through the intestinal cell through endocytosis, and then it'll merge, you know, with these vesicles here, and it'll be carried all the way through that cell to the other side, and then it will be absorbed through the villi, the finger-like projection, into a lacteal or a blood vessel, depending on what type of nutrient it is. Plicae circularis are going to be those folds of the mucosa wall. So you might be thinking like, what is diverticulitis? And so we'll get to that with the colon. But if you were thinking that, you're kind of on the right track, because diverticulitis is going to be out pockets of the mucosal wall of the colon. Okay, so secretions of the small intestine, as you've guessed, mucus, as we know, watery fluid, and then enzymes. So let's look at those enzymes. Peptidases are going to break down peptides into amino acids, so more protein synthesis. We have carbohydrate enzymes, so we've got sucrase, maltase, lactase. So sucrase is going to break down sucrose, maltase will break down maltose, and then if you have it, lactase will break down lactose. And then we have lipase, which will break down fats into fatty acids and glycerol. So here is the summary of the major digestive enzymes. And I love this because even though we're in the intestine now, it includes your salivary glands and your chief cells of your stomach, and then also the pancreas and the intestinal mucosal cells. So there's your one-stop shop, table 17.9, for you to go back and review for your digestive enzymes. Regulation of small intestinal secretions. So the regulation occurs by these methods. Mucus secretion occurs in response to mechanical stimulation or the presence of irritants such as gastric juice. Contact with chyme stimulates goblet cells to secrete mucus and intestinal glands to secrete watery fluid. Distension, remember the stretch of the intestinal wall, will activate nerve plexuses in the wall of the small intestine. And distension, that stretch, will also stimulate parasympathetic reflexes that trigger the release of intestinal enzymes. Absorption. So this is a biggie because remember, this is the main site for absorption in all the digestive system. So villi are going to increase the surface area of absorption. The small intestine is the most important absorbing organ. Absorption is so effective that very little absorbable material reaches the organ's distal end, particularly if you have a healthy small intestine. Malabsorption is when you're not able to absorb material. It can cause diarrhea and all kinds of stuff. Leaky gut is when things can leak into your body from your intestines. So we need very little absorbable material to reach the distal end of this organ if we want complete health. All right, so carbohydrate digestion and absorption. Salivary and pancreatic amylase will break down starch and glycogen into disaccharides. Intestinal enzymes will break down disaccharides into monosaccharides. And then monosaccharides are absorbed by facilitated diffusion and active transport into the blood vessels in the villi. So carbohydrates can go into the blood straight. Protein digestion and absorption. Pepsin in the stomach breaks down proteins into polypeptides. Pancreatic proteases such as trypsin and chymotrypsin break down proteins and polypeptides into smaller peptides. Intestinal peptidases break peptides into amino acids, and the amino acids are absorbed by active transport into the blood vessels of the villi. So both carbohydrates and proteins can be directly absorbed into the bloodstream, which makes sense because these are polar molecules, and so they can interact with water, which blood is mostly water. Figure 17.39 and 17.40 shows a disaccharide and a dipeptide. So the disaccharide here is maltose, so that's going to have two glucose molecules, and the enzymes are going to come in. We see maltase here is going to bring hydrolysis into the picture, and we're going to add an H and an OH group in between our maltose molecule, and that will break it up into the two glucose. That's called hydrolysis. So then dipeptidase is going to do that with a dipeptide from protein digestion, and it's going to break it by adding water. So again, hydrolase, and it's going to bring in the OH and the H. That's why it's called a hydrolysis. Lysis is to cut. Hydro is using water, so we're going to use water to cut it. So we add a H2O, hydrogen and hydroxide, and we cut that amino acid down into its two subunits. Now, fat digestion absorption is going to be a little bit different. Remember, it's going to need bile salts, and then digestion mainly by enzymes from the pancreas and the small intestines, digested into glycerol and fatty acids, and these are going to have to go into the lacteals, the lymphatic capillaries. And then micelles and chylomicrons are going to be very important in this fat absorption and digestion. So micelles are loose complexes of fatty acids and bile salts, from which fatty acids can migrate to microvilli and be absorbed. Fatty acids plus glycerol are resynthesized into triglycerides in the smooth ER. New triglyceride clusters are encased in protein, forming chylomicrons, and the chylomicrons are absorbed by the lacteals. Fat is not soluble with water. Remember, it's a nonpolar substance, and so it has to be broken down by bile salts, and then it has to be packaged into the micelles and chylomicrons in order for that fat to get into the bloodstream. And of course, we give fat a bad rap because if we eat too much of it, then it can cause high cholesterol, high triglyceride levels in the blood, high LDL levels, etc., and can lead to heart disease and cancer. But without fat at all, then, we would lose a very important source of energy. Our cells really utilize fat for energy. And also, fat is important in, not only energy, but shock absorption, the menstrual cycle. Without enough fat, a woman's menstrual cycle would cease, and we would be very malnourished. So the nervous system would suffer. There's a lot of fatty tissue in the nervous system. All right, but I digress. Figure 17.41 shows fat digestion. And so as you can see, we've got to use lipase to break down the fatty acids into the 3 fatty acid chains and the glycerol. And then it'll be packaged into the chylomicrons as mentioned here. So number 1, fatty acids resulting from triglyceride digestion enter an epithelial cell, And number 2, fatty acids are used to synthesize triglycerides in the endoplasmic reticulum, the smoothie R. Number 3, triglycerides and other fats are collected in clusters encased in a protein to form chylomicrons. Number 4, chylomicrons leave the epithelial cell and enter a lacteal. And number 5, lymph in the lacteal transports chylomicrons away from the intestine. And when we get to the lymphatic system, you'll see that the chylomicrons and all that are just kind of going ahead and dump like right behind the thoracic duct in your chest. Like, oh, we'll just dump that fat right there behind the heart. All right, so table 17.10 is going to summarize the intestinal absorption of nutrients. And here is a review of the movements of the small intestine. So, of course, you have peristalsis, which is that wave-like pushing movement. So go back to that sock. You're pulling that sock and squishing it. And then also segmentation is like the chewing. It's a mixing movement that further mechanically digests food in the small intestine. And then parasympathetic is stimulating of the small intestine, and sympathetic would inhibit. And then we see that if the small intestine becomes over-distended or irritated, a strong peristaltic rush may sweep contents quickly into the large intestine, and that can result in diarrhea. I'm sure we've all felt that a time or two. And the ileocecal sphincter joins the ileum of the small intestine to the cecum of the large intestine and helps regulate the flow of chyme. So now it's time to go to the large intestine. So this is your colon. And this is a very important organ because, of course, we know that cancers can form here and all kinds of stuff. So organ health overall is important. And, you know, maintaining the health of your stomach lining, maintaining the health of your small intestine will all benefit the large intestine and your body overall. And again, it's a day-to-day struggle. I ate more homemade pancakes today than I ever should have, you know, and luckily they were homemade, so I knew exactly what ingredients I had put into them. But, you know, to balance it off this evening, I will be having lean meat and vegetables. And so again, we just try every day to make mindful choices to keep our intestines and our bodies happy. So the way that the large intestine works is because it's going to absorb water and electrolytes, and it's also going to reabsorb and recycle water and digestive secretions, and it's also going to form and store feces. And then that first bullet reminds you that it's named the large intestine because the diameter is greater than that of the small intestine. It's way shorter though. See how it's like 1.5 meters long? So, you know, it's not even half the length of the small intestine by a long shot. It's just wider. So like I said, the small intestine should be called the narrow intestine. The large intestine should be called the wide intestine, but whatever. All right, so here are the parts of the large intestine. We've got the cecum, the colon, the rectum, and the anal canal. The cecum is pretty cool. A lot of animals have the cecum. We have a reduced cecum because of us being omnivores. If we were like herbivores, we would have a really large cecum that would be filled with microbes that help us digest cellulose from plants. But we don't. So what we have is a reduced cecum that has a little appendix attached to it. So that's a structure that came from, you know, maybe herbivorous ancestry, but we no longer need that. And so the appendix is going to be a blind sac attached to the cecum. It does have lymph nodules in it, and it functions in the immune response. But unfortunately, because it doesn't really function for anything else, it can just get swollen and infected, and that, of course, is appendicitis. The colon itself is going to consist of 4 parts. You might not have known this. It has the ascending, which goes up. It has the transverse, which goes across. It has the descending, which goes down. And then it has the sigmoid, which is an S shape. And it contains the hepatic or right colic and sphenic left colic flexures. Then the rectum. The rectum extends from the sigmoid colon to the anal canal. It lies next to the sacrum, and, you know, it stores the feces before it is emptied out the anal canal. So the last 2.5 to 4 centimeters of the large intestine is the anal canal. Opens to the outside as the anus. You've got 2 sphincters. Thank the Lord. You've got an internal and an external. So you got two ways to hold it in. And so the internal is involuntary, and the external is the one that you can squeeze to help keep you from going before you get to the bathroom. And so here we see in figure 17.43 the parts of the large intestine. Here's the little appendix that attached to our cecum. So remember, our cecum is going to be vastly reduced because we're omnivores, not herbivores. And so the appendix would be where, like in an herbivore, it would have extended out. Sometimes they go all the way out and they spiral around like in a koala bear. It's really cool looking. Anywho, uh, in a rabbit, the cecum ends up looking like a carrot. It's so funny. I'm like, really? So yeah, herbivores have really different looking colons than we do. All right, but let's get back to the human. So we have the ascending colon, which travels superiorly towards the head end. And then we have the transverse, which is named because it goes horizontally. And then we have the descending because it goes inferiorly away from the head. And then we have the sigmoid colon, which is an S-shaped. And then here's the rectum, which stores the fecal matter. And then the anal canal is going to have the two anal sphincters, and poop will be expelled through the anus. Here we're seeing the large intestine, rectum, and anal canal. And these kind of structures that you see are called haustra. Hopefully we'll talk about those in a second. And then look at the external anal sphincter and all those blood vessels in there. Hemorrhoids are going to occur when we're, straining too much, and the blood vessels get stretched out and inflamed around the anus in the anal canal. You can have internal and external hemorrhoids. All right, so here we get to learn about the haustra that I just mentioned. So the wall has the same four layers as the other tubular organs of the alimentary canal. So that would be the mucosa, the submucosa, the muscularis, and the serous. It does not have villi or plica circularis. It does have longitudinal muscles, though, that are organized into three bands. The tenia coli that run down the entire length of the colon, and muscle bands create pouches called haustra, which help form feces. So haustra. And sometimes when you see the poop, if it's kind of got those little pouchy-like looks to it, you can see it's from those haustra. And here is the mucosa, submucosa, muscularis, and serosa. So the large intestine has little or no digestive function. It contains tubular glands called with goblet cells in it that secrete mucus. We're still in a mucous membrane, and it's the only significant secretion of the large intestine. The large intestine absorbs water, about 90% of the water that enters it, and that is so important. Otherwise, we would, dry out and become dehydrated. It also absorbs electrolytes. It houses intestinal flora, which are bacteria that break down contents such as cellulose, which, in a herbivorous animal, like that's why they have the cecum, because they need a lot of microbiota to help them break down cellulose. In turn, these microbiota will produce vitamins K, B12, and thiamine. The large intestine will form feces and carry out defecation. And this is where your gas is going to back up. So it's a mixture of nitrogen, oxygen, methane, carbon dioxide, hydrogen, ammonia, and hydrogen sulfide, which is going to be part of that unpleasant odor. And a lot of that comes from the microflora that are in your colon that are working on your food and breaking it up. And the gases are the byproducts of their metabolism. So here we see the large intestine mucosa with the goblet cells, and then of course there's that lumen. Movements of the large intestine are similar to those of the small intestine. However, they are slower and less frequent than those of the small intestine. There's a couple of types of movements. There's mixing movements, and it looks like they'll call them the mass movements and the defecation reflex. So we've got, the peristaltic waves, of course, will be 2 to 3 times a day, which usually follow meals. And then the defecation reflex. This eliminates feces from the body, and it involves holding a deep breath and contracting the abdominal muscles. Feces move into the rectum. Peristaltic waves then occur in the descending colon, and it relaxes the internal anal sphincter and then the external anal sphincter. Feces are composed of materials that are not digested or absorbed, and also it contains water, electrolytes, mucus, bacteria, and the bile pigments, which provide the color after bacterial alteration. The pungent odor is produced by bacterial compounds and can include phenol, hydrogen sulfide, indole, skatole, and ammonia. If you remember, hydrogen sulfide is going to smell like eggs. Disorders of the large intestine. So here's that diverticulosis, which I had said you might be thinking about earlier. This is pretty severe. It's a weakening of the intestinal wall, and it can lead to the protrusion of the mucous membrane. It forms outpouchings that can become inflamed and infected. And then we have inflammatory bowel disease. So this is a group of disorders including ulcerative colitis. This affects the mucosa and the submucosa of the large intestine, and it causes bloody diarrhea and cramps. And I believe ulcerative colitis can also be like an autoimmune disease, so can like Crohn's disease. Crohn's disease is more serious as it affects all layers, and it occurs in both small and large intestines, and it causes diarrhea and pain. And many colleagues that I've known who have had like Crohn's disease throughout my years, they have ended up having to have portions of their intestines removed. And then colorectal cancer. This is the cancer of the large intestine or the rectum, and it's the fourth most common cancer in the United States. And screening tests include fecal occult blood testing and a colonoscopy. And so it's time to start getting your colonoscopies around like the age of 45 to 50. And, they're even starting to let patients take it even earlier if they have a family history. Last but not least, some lifespan changes of the digestive tract. So changes to the digestive system are slow and slight and eventually include the following. The tooth enamel thins and teeth may become sensitive. Gums may recede. Teeth may loosen, break, or fall out. The GI tract becomes less efficient. Slowing peristalsis may lead to heartburn. Gastric secretion slows. Constipation may become more frequent. Nutrient absorption decreases, including fat-soluble vitamins. Incidence of lactose intolerance increases, and accessory organs age, but typically not necessarily in ways that affect health. And that is a wrap. Thank you all for listening.